The next topic we're going to deal with is the juxtaglomerular apparatus. The juxtaglomerular apparatus. We see things we already know here. This is glomerulus with Bowman's capsule. Afferent arteriole coming in here, so blood's coming in this direction, goes through the glomerulus, then comes out through the efferent arteriole heading away from the glomerulus. Adjacent to this the vascular pole of the glomerulus and the renal corpuscle here, we have one loop, one section of the distal convoluted tubule. So this is distal convoluted tubule. The fluid that's passing through this distal tubule is what's the beginning of our urine content. In here we've had a lot of the material removed from that fluid, the sugars, many of the ions, most all of the water. So this is a concentrated product coming through here. So anyway, this has to come up right next to the base of the glomerulus there, and there's a interaction between the cells along this side of the distal tubule and the cells right here at the last part of the afferent arteriole. Now these cells down here, these are smooth muscle, as you would find around any arteriole. The next group of cells are kind of a modified version of smooth muscle. They kind of change a little bit and they become primarily hormone producing cells. These cells right in here are referred to as juxtaglomerular cells. Juxta means next to, of course. So there are juxtaglomerular cells right there. The cells in this region of our distal convoluted tubule, their nuclei tend to be much closer together. This compact zone of nuclei along this wall makes this location, a particularly special location, we refer to as the macula densa. Macula densa. Macula refers to a spot. Densa just means they're dense. So it's a dense place where our nuclei are more concentrated, not as spread out as they would be along the rest of our distal convoluted tubule. These are all normal distal convoluted cells out here, so you really don't see the separation between the cuboidal cells out here in the distal tubule very well, but you can see the spacing of the nuclei very well. So those are normal cells of the distal convoluted tubule. These are not. These are special cells of the macula densa. They're smaller cells, more tightly packed nuclei along this wall. So there's an interaction here that's very important to regulating blood pressure, and we're going to talk about that next. So here's your juxtaglomerular apparatus, this interplay between macula densa and juxtaglomerular cells at the vascular pole of your renal corpuscle. Now we're going to look at the role of the juxtaglomerular apparatus and how it helps to control blood pressure. So the first thing here, if we have a lower blood pressure, then the flow of fluid into our glomerulus is going to be less. If there's not as much pressure there, then we don't have as much fluid moving through the wall of the glomerulus into Bowman's capsule and into the nephron, the proximal tubule, the loop of Henle and distal tubule. So we have a slower movement of that fluid going through there. Those cells of the nephron are going to have more time to retain all of the solutes and things that you want to retain in your body, sodium being the important one of concern here. So if you have more time to absorb sodium, there's less sodium in the fluid that reaches the macula densa in there, so low blood pressure leads to a lower sodium level, and the macula densa responds to that low levels of sodium in the fluid moving through the distal convoluted tubule. These cells are specially modified to release a kind of paracrine substance. paracrine substances are like hormones that instead of going into the blood and circulating like hormones do, they are released to their neighboring cells. They're a chemical stimulant to the neighboring cells to convey some kind of information that way. So like a hormonal control, but only to its neighboring cells. So paracrine stimulation of these juxtaglomerular cells that are just next to it lead to the release of renin. So juxtaglomerular cells around that afferent arteriole will release renin when stimulated by low blood pressure and paracrine hormones of the macula densa. So next we're going to look at the role of renin in what's called the renin-angiotensin pathway to retain our blood pressure, get our blood pressure back up. All right, here we have the release of renin, remember, from the juxtaglomerular cells that wrap around the afferent arteriole of the renal corpuscle. So this chemical is released into the bloodstream. It has its effect on a protein that is present in the blood, that is circulating in the blood, produced by the liver primarily, angiotensinogen. Angiotensinogen. So the angiotensinogen is converted to angiotensin 1 in the blood. This conversion leads to the next step, which is the formation of angiotensin 2. This is done by angiotensin converting enzyme, also known as ACE, ACE. We've probably heard of that before. Angiotensin converting enzyme that's produced mainly in the lung. This is where, at least where most of this conversion occurs, in that vascular circulation through the lungs. Angiotensin 1 is converted to angiotensin 2. Angiotensin 2 has a role in vasoconstriction. It's a general vasoconstrictor. It causes constriction of your blood vessels, and that can lead to an increase in blood pressure if your blood vessels constrict. Also, angiotensin 2 causes the release of aldosterone from the adrenal cortex. This aldosterone causes water retention in the kidney. It increases the absorption of water across the wall of your loop of Henle and your other tubules, and largely in the collecting ducts. A lot of water reabsorption across the collecting ducts in the kidney. So if you don't lose fluid, it helps you to retain your blood pressure, right? So you need fluid in there. And the only way to actually bring everything back to normal is to intake more water eventually. Get more volume of water into your system. So you have to do something to get this system to break its cycle. So if you get enough water, get things back to normal, you're going to inhibit renin release. So if your blood pressure goes back up, you have more water in there, more circulation through the glomerulus. That's going to lead to the lack of stimulation of juxtaglomerular cells by the macula densa, and you'll return to a normal state there. And normal kidney function, as well as normal blood pressure. So, go through it again. Renin causes angiotensin in the blood to convert to angiotensin 1. Well, I should say angiotensinogen in the blood to convert to angiotensin 1. As it circulates through the lung, angiotensin converting enzyme converts that angiotensin 1 to angiotensin 2. Angiotensin 2 is a vasoconstricting chemical. It causes vasoconstriction, and it causes aldosterone release from the adrenal cortex. Aldosterone causes water retention, so you don't lose as much water. And both of these things, retaining your water and constricting your blood vessels, leads to an increase in your blood pressure and returning of the normal flow through the kidney. Now for a look at the nephron tubule system. Over here we have the proximal convoluted tubule. It leads into the loop of Henle that descends down into the medulla of the kidney, and it comes back up to the cortex. And we have the distal convoluted tubule that leads into the collecting duct over here on this side. We want to talk a little bit about the histology of these cells that make up the look of the cells that make up the proximal tubule, the loop of Henle, distal tubule, and collecting duct next. There are more proximal convoluted tubules, well, not more tubules, but more length of proximal convoluted tubule in the cortex than there is distal convoluted tubule, and we'll talk about that here in a minute. Let's first look at the overall structure of the cross-section of a proximal convoluted tubule. So let's say this is the cross-section of the tubule here. On the inside of the cells, the margin is kind of obscured because they have a number of microvilli out here. These are highly absorptive cells, so you expect them to have all these microvilli kind of obscuring the luminal space inside that cross-section. These are simple cuboidal cells, and we can see the arrangement of nuclei around here. A very regular occurrence of nuclei, round nuclei, kind of evenly spaced. But we can't see the separation of the cells. There's no line separating one cell from the next. That's because there's an extensive interdigitation, and I'll draw this in here, though you don't see it through the microscope. There's an interlacing of the cell bases here between two cuboidal cells. So that kind of obscures where one cell stops and the next cell begins. It all looks kind of continuous on through there. So that's what you see in a proximal convoluted tubule. No separation of the cells by straight lines in there, and crowding of the lumen by all these microvilli there. So there are lots of these profiles, cross-sections of proximal tubules out there in the cortex. Next, the loop of Henle. As we drop down into the loop of Henle, the tubule gets really, really small. Now down here you're going to be reabsorbing a lot of water. Water's coming out of this tubule as it circulates through. The cross-section of a loop of Henle is quite different. It's going to be smaller overall. It's going to have a very clear center, and you have low cells, and then you have a nucleus bulge out, and then you have low, then a nucleus bulge out, and then it'll be low, and a nucleus will bulge out. So you'll have a very tiny cross-sectional area of the space in there, and here's a nucleus here, a nucleus here, a nucleus here, around the wall of that loop of Henle in there. Much smaller than the cross-sectional area of the proximal and distal tubules. Just two or three nuclei typically around that luminal space, which is usually more collapsed even than what I've drawn in my picture. As we head into the distal tubule, they're about the same size as the proximal tubule. They also interdigitate at the margin, so it's a little bit hard to see the separation of individual cells, but they don't have this crowding of microvilli on the inside. So you usually see a fairly discrete, it may be a little bit fuzzy, but not too bad. You have a nice circular lumen or cavity on the inside of a distal convoluted tubule, and then your nuclei, let me get this right, they're kind of pushed away from the basement membrane. They're closer to the surface, toward the luminal surface, something like this. So when you're looking at the cortex of the kidney, it's fairly easy to tell which of the tubules you're looking at that are proximal convoluted tubules and which ones are distal convoluted tubules. When we get to the collecting duct consists of a more columnar cell type than what we see in the other portions. And of course it's going to be larger, it's going to be carrying more fluid. So I'll make a collecting duct here. And the margins, those edges of the cells can be discerned now. So we can see individual cells with straight edges. That interdigitation at the margins no longer a factor for us, so we can see straight-edged cell boundaries. The lumen has a sharp boundary to it. Now in the upper portions of the collecting duct, they may be only cuboidal instead of columnar cells, but as you go down, they become more columnar. Even the upper portions of the collecting ducts, you can still see that separation, those clear margins at the, where one cell meets another on their lateral surface in there. So, proximal tubule, loop of Henle, distal tubule, and collecting duct. They look different under the microscope, and we'll see that in lab. What I've drawn here is the end of a couple of collecting ducts as they come toward the renal pelvis. They open up at the what's called a renal papilla. A renal papilla. It's a little bump that extends out into a minor calyx. So this would be the space of a minor calyx here. This is our renal papilla, this region right here. Renal papilla, just a little bump, sticks out. And in that bump, there are all these pores from all the collecting ducts that converge here. Remember, there's a renal medulla where all of the, it's a renal pyramid, where all of the collecting ducts kind of come to a point, and this is the point that they come to in the minor calyx, that renal papilla there. This area right here that has all the little holes in it from all of the different collecting ducts that come down here is also known as the area cribosa. That means it has a bunch of holes in it. It's basically what that means. Anything that's cribiform has tiny holes in it, like perforations, like a colander kind of arrangement. But anyway, tiny holes in this bump from all the tubes that come in here. Now the epithelium on there was a simple columnar epithelium, and that columnar epithelium kind of lines all of this space. And on the proximal side of the near side, I guess you'd call it, of the kidney pelvis, you have mostly a simple columnar or simple cuboidal type epithelium. But on the far side, as you approach the renal, I guess the ureter, on that side of the renal pelvis, on the far side of the renal pelvis, it converts from a simple epithelium to a transitional epithelium. So the beginning of your transitional epithelium that runs through your ureters, urinary bladder, and urethra actually begins in the renal pelvis near the connection to the ureters down there. So we're going to look at a picture of the area cribosa on a renal papilla and electron micrograph in your textbook. So these two images, the one above and the one below, are both of a renal papilla. The papilla is real obvious in the upper picture in the electron micrograph. It's a scanning electron micrograph of a renal papilla, and you can see the tiny pores indicated by the arrows in the picture. So that's where all those collecting ducts enter into a minor calyx of the kidney. The lower picture is a cross section of a normal light micrograph. And at the very bottom there, you can see where those collecting ducts enter into the space down below. Now, on the opposite side from that, this side over here, it's going to be all transitional epithelium. On the other side, near the renal papilla, up there in the area cribosa, it's going to be all simple columnar and simple cuboidal epithelium.